Welcome to today's webinar titled Metabolic Disrupting Potential of Unconventional Oil and Gas Operation Chemicals and Mixtures. I'm Kim Schultz, the Oil and Gas Program Coordinator with TEDx, the Endocrine Disruption Exchange. TEDx is a science-based nonprofit research institute, and our mission is to reduce the use and production of chemicals that interfere with healthy hormone function. This webinar is part of an ongoing series inviting scientists to present their research addressing health effects associated with unconventional oil and gas development. We welcome any comments or suggestions. Contact us by going to our website at tedx.org. It is my pleasure to introduce our speaker today, Dr. Chris Kassotis. Dr. Kassotis is an NRSA postdoctoral research fellow in the Nicholas School of the Environment at Duke University. He completed his PhD at the University of Missouri, working with Susan Nagel to assess unconventional oil and gas operations as a novel source of endocrine disrupting chemicals in water and the potential for adverse human and animal health outcomes from exposure. He is now working in Heather Stapleton's lab at Duke to assess the adipogenic activity of complex chemical mixtures present in indoor house dust and other matrices, as well as to develop new assays to isolate and characterize novel endocrine disruptors and complex mixtures. Thank you so much, Dr. Kassotis, for joining us today and presenting your research, and I will now turn it over to you.
Thanks so much. All right, let me share my screen here. All right, hi everyone. I want to thank the folks at TEDx for allowing me to come and share some of my recent work with you today. As Kim said, I've been a postdoc at Duke now for about 3 years and continued working on projects related to fracking and really stepped into a world of, into a different world of environmental mixtures related to the indoor environment, that of indoor house dust. So specifically, I'm interested in metabolic disruptors or environmental contaminants that could be contributing to the chronic metabolic health trends in humans, including obesity, diabetes, et cetera. And given my research here on adipogenesis, the process of fat cell development, it was an obvious question to ask whether there were metabolic disrupting chemicals present in unconventional oil and gas or fracking fluids, and whether these might influence fat cell development. So moving to slide 2, the outline, I'll just give a brief description of an unconventional oil and gas operation, chemicals, and fracking. Introduce hormones and endocrine disrupting chemicals, and then introduce adipogenesis, or the process of fat cell development. From there, I'll move into describing our recent publication from Science of the Total Environment, and then discuss some results and next steps. So I'm sure nearly everyone at this point is familiar with this popular ProPublica graphic. Very generally, unconventional oil and gas production involves the coupling of 2 technologies. The first, horizontal drilling, where industry is able to drill down thousands of feet to a shale or coal bed gas layer, and can then turn the drill bit horizontally and drill through that target geological layer for some distance, in up to 2 miles in some cases. This is then coupled with hydraulic fracturing itself, which involves the high pressure injection of millions of gallons of water mixed with sand and proppants into the well. The force serves to fracture this layer. The proppants, which are typically finely grained silica sand particles, prop open these fractures, and the trapped oil and gas particles are able to flow out of the layer and be collected. So on the left of this slide, you have the typical proportions that we think of for hydraulic fracturing. The total injected mass is typically around 7% proppant, 91% water, and about 1% of a chemical mixture, though this graphic focuses on the liquids only. That may seem like a small amount of chemicals, though with an average injection volume of several million gallons, even 1% is a very large volume of chemicals going into each and every well. On the right, this is just to highlight some similar techniques, including matrix acidizing and acid fracking. And I just wanted to highlight that these have a considerably higher total proportion of chemicals in the injected mixture. So this graphic we adapted with permission of the Checks and Balances project, and it really highlights many of the pathways associated with potential fracking contamination of water. So surface water can be impacted by surface spills during the transportation, fracking, and recovery processes, through leaking wastewater storage pits, in some cases unlined. Faulty cement well casings have been shown to be a concern in several studies and are a path for contamination of groundwater, along with surface spills. And lastly, there's a lot that we still don't know about the fractures occurring underground, and whether there's potential for migration of these fluids over time into the groundwater in a particular region. So this slide just highlights some of the data that we do have on oil and gas operation spill rates. While some studies have suggested lower rates, closer to 2%, which you'll see have been true in certain regions and in certain years. In other regions and other years, you have rates that approach 20%. So it's really quite variable across regions and across time. So we were spurred into this research area through the work of Theo Colburn and the folks at TEDx. This 2011 paper assessed 353 chemicals that could be identified at that time by CAS number, and found that as many as 37% of these had some suggestion in the literature that they might be endocrine disrupting chemicals. So that spurred our interest. So what exactly is an endocrine disrupting chemical? So if we step back for a moment, hormones are essentially the chemical messengers of the body, regulating numerous processes during development and maintenance throughout our lives. Pictured here are 5 well-described hormone receptors, and this is just meant to give you an idea of the vast number of developmental and maintenance processes that these hormones regulate. Also, it's meant to communicate how there's a lot of overlap between these systems. It's rarely one hormone that regulates a single process in the body. Slide 9. So the Endocrine Society defines an endocrine disrupting chemical as a chemical or mixture of chemicals in the environment that interferes with any aspect of hormone action. This disruption can occur through 2 main mechanisms. One, direct interactions. In this case, we have chemicals able to mimic the actions of endogenous hormones, acting as receptor agonists. So they can bind to hormone receptors and drive a receptor-mediated response. Antagonists, in contrast, can bind to hormone receptors and prevent the endogenous hormone from binding, and thus prevent that biological response. The other main mechanism is through indirect interactions, and this can occur through a variety of different mechanisms. Endocrine disruptors can influence where and how frequently receptors are expressed in various tissues. They can modulate hormone levels in the bloodstream, and they can influence how a receptor responds to its natural ligand or hormone, among other mechanisms. So ultimately, these chemicals can disrupt the developmental and maintenance processes I described before and can lead to a host of adverse health outcomes. These have all been described in controlled animal models, and most, if not all, have been associated with endocrine disrupting chemical exposure in human epidemiological studies. One that I want to highlight here is metabolic disruption, as metabolic function is intimately connected and regulated by a suite of hormones. So in the interest of time, I'm not going to rehash all of our previous studies describing unconventional oil and gas chemicals as endocrine disruptors. However, I will highlight some findings from our developmental exposure work in mice, both in our laboratory and through the work of our colleagues. What I want to highlight here is that gestational exposure to a mixture of 23 commonly used hydraulic fracturing chemicals was able to induce weight gain, increased birth weight, and increased weight into early adulthood for both male and female mice. This suggested to us that there might be some metabolic perturbations to assess more closely. So this idea that environmental chemicals could be playing a role in this, this trend of worsening obesity and overweight was first posited in this 2002 publication. Though importantly, this paper really summarized decades of experimental evidence at that time. So in this paper, the authors challenged caloric intake and activity as insufficient to account for the magnitude of the change in obesity prevalence, and genetics as insufficient to account for the rate of the change. They also summarized the wealth of available animal evidence on antibiotics, PCBs, plastics, pharmaceuticals, and other chemicals that have all been demonstrated to increase weight or fat mass in animal models. So if we look at the development pathway for a fat cell or adipocyte, this is what we have. If we start from a mesenchymal stem cell precursor in the upper left, we have a multipotent cell that can develop down several pathways into fat, bone, or other cell types. So the next step down the development pathway is these fat precursor cells shown in the red box and on the small inset graphic to the right of that. These are cells that are committed to the fat cell lineage. They can't develop into other cell types at this point, but are very early in development and require signals to develop fully into what we would think of as a mature adipocyte or fat cell. This is the stage that most cell models target. And activation of several of these early stage markers, such as the nuclear receptor, peroxisome proliferator activated receptor gamma, or PPAR gamma, can push development forward. As that occurs, the cells change from a fibroblastic morphology, shown at the top, undergoing changes to a more spherical cell type and accumulating lipids or triglycerides around the periphery of the cell, as shown on the bottom right panel. And then as they become mature fat cells, these lipid droplets coalesce and become a large central lipid droplet that displaces the nucleus and eventually resembles a mature human white fat cell, as shown on the bottom left. Importantly, while PPAR gamma is considered the master regulator of this process, activation of other receptors can also promote the process, including the glucocorticoid receptor, retinoid X receptor, and others. So before I get into the study itself, I want to explain our cell system a little bit more clearly. So we use 3T3L1 cells. These are committed fat precursor cells, like I described in the previous slide. Swiss albino mouse embryonic fibroblast cell line. So they cannot develop into other cell types, but they do require a push to develop into mature fat cells. This model is quite well understood as it's been in use since the 1970s. And so we understand the mechanisms involved with it, and it's proven reliable, particularly when coupled with assessing the underlying mechanisms, such as PPAR gamma, in predicting in vivo health outcomes. Some limitations of the model are that it does take about 2 weeks to perform each assay. The cells have a limited lifespan, and it fails to incorporate some of the other mechanisms that can contribute to metabolic dysfunction in a whole organism. So in general, our assay timeline looks like this. We take the pre-adipocytes and plate them into 96-well plates and let them grow to confluence, or so that there isn't any additional room to grow in the plates. This is a trigger for the cells to undergo growth arrest and revert to clonal expansion, which is a prerequisite for the cells to differentiate into mature fat cells. So once they've spread across all the space in the walls of the plate, we give them a further 48 hours to do that. And after that, we'll give them a push down the adipocyte development pathway by treating them with what we would call a differentiation cocktail. This includes fetal bovine serum, insulin, which increases the rate of lipid accumulation and allows it to be a 2-week assay as opposed to a month or more, and IBMX, which is a PPAR gamma agonist. We also start treating with our test chemicals, whether that be water samples or chemicals or anything. And we do that for 48 hours. And that's that shown in that red box there. At that point, we remove the IBMX and continue treatment with everything else for a further 8 days. Removing that PPAR gamma agonist allows us to establish a baseline. It pushes the cells towards differentiation, but we get minimal lipid accumulation in our solvent control wells. And that allows a greater dynamic range to assess lipid accumulation by our positive control, which in this case we use rosiglitazone, and our test chemicals. So at the end of that 2 weeks, we then test for lipid accumulation using the Nile red dye, shown on the inset figures in the middle, which partitions into the lipid droplets and fluoresces, and also for cell proliferation using a HESH DNA dye. So from here, I'll transition into our recent study titled Unconventional Oil and Gas Chemicals in Wastewater Impacted Water Samples Promote Adipogenesis via PPAR gamma dependent and independent mechanisms in 3T3L1 cells, which was recently published in Science of the Total Environment. So we selected the 23 mix of common unconventional oil and gas chemicals that I described earlier and is shown on the inset table here. Several unconventional oil and gas wastewater samples collected in Colorado, and several surface water samples purportedly impacted by unconventional oil and gas operations from previous excursions by our lab. So 4 from Colorado and 4 from West Virginia. And we evaluated all of these samples and mixtures across a range of concentrations for their abilities to promote fat cell development and also to assess the putative mechanisms that might be driving those effects. So to describe how I'll be showing my results, here's a 2-panel figure. So if you focus first on the blue line, this shows my positive control rosiglitazone, which is a pharmaceutical used in diabetes treatments. So on the left, you'll see the triglyceride accumulation data. So an increase in lipid content within a well could be due to either individual cells incorporating more lipids or a greater number of cells with the same lipid content. So by normalizing lipid content to DNA, we can consider lipid accumulation per cell, which is what's shown here. You'll note that rosiglitazone, which again, to remind you, is shown in blue, promotes a robust response with increasing concentration along the axis and reaches maximal lipid accumulation around 1 micromolar. This maximum is then used to set the ceiling of the assay, and all other test chemical responses are presented as a percent of that maximal positive control response. On the right is the pre-adipocyte proliferation data, or DNA content. And everything is relative here to the vehicle control. So this only receives the differentiation cocktail, so the push towards the mature fat cells, and then the solvent that the test chemicals or samples were diluted in, but no other treatments. So anything greater than 0 on the y-axis is an increase in DNA content, or pre-adipocyte proliferation, as we would view it. And anything below is what we would term cytotoxicity. Here you can see about a 20% increase in DNA content at the high dose of our positive control. Now, if you take a look at the red bar, which is the 23 mix of common unconventional oil and gas chemicals that I'd mentioned previously, it induces a fairly modest effect on triglyceride accumulation, or lipid accumulation, on the left. So you see a weaker response and at higher concentrations than the positive control. But induces substantial pre-adipocyte proliferation, so much greater effects than the positive control. Slide 18 here. So this is a bit busy, so we'll take a minute to ensure it's all clear. So first, the x-axis is set equal to relative water concentrations. So 0 is essentially undiluted or unconcentrated water. -1 represents a 10-fold dilution of water, and +1 represents a 10-fold concentration of water. So what you may first notice is that most of the activity here is occurring at concentrations less than 1x, or less than that of pure water. And second, what is harder to see is that there are several samples here that have no effects on either measure of fat cell development. And that is the Colorado process control and the West Virginia blank. Both are control samples included to ensure no lab or sampling induced effects, and they're both negative. West Virginia 2 is also negative for both measures and is a reference stream nearby a wastewater injection well site where West Virginia 6 and 3 were collected. And so that reference stream is also negative. In contrast, the rest of the samples all promoted some degree of activity. So wastewater 1 induced very strong effects on both triglyceride accumulation and pre-adipocyte proliferation, while the others promoted more modest effects, and only in some cases on pre-adipocyte proliferation. Notably, wastewater 1 induced effects at a 100-fold dilution of the wastewater, suggesting it could potentially induce effects even when diluted into receiving waters. And the other samples, some of the surface water extracts from Colorado and West Virginia, induced effects at concentrations of about 1 to 25 dilution of fresh water. So I previously mentioned that activation of PPAR gamma is one of the main mechanisms through which contaminants can induce fat cell development. So as such, we screened all of these mixtures and samples through an assay to assess PPAR gamma activation, and found that many of the samples exhibiting significant effects on lipid accumulation or pre-adipocyte proliferation also activated PPAR gamma at similar concentrations. However, several of the samples, notably Colorado 15 and West Virginia 6, appeared to exhibit effects through a mechanism other than PPAR gamma. So in an attempt to better delineate what those other mechanisms might be, we've previously characterized the 23 mix of chemicals and these surface water extracts for their ability to act as agonists or antagonists for 5 other hormone receptors. So highlighted here are results of antagonism of these 5 receptors. And 2 that I'll highlight are the androgen and thyroid receptors. So inhibition of both of these pathways have been demonstrated to promote fat cell development and could be then other molecular mechanisms driving these effects. And all that I'll say here is that the samples lacking PPAR gamma activity do demonstrate inhibition of these pathways, suggesting that these mechanisms could be causative mechanisms through which some of the fat cell effects are being promoted. So just a brief foray away from this study, we've actually done some work to assess potential candidate chemicals that could be inducing some of the effects observed in fracking fluid. One of these candidates is the alkylphenol and alcohol polyethoxylates. So these are high production volume chemicals used widely in laundry detergents, hard surface cleaners, paints, cosmetics, agriculture, and more. As such, they're common environmental contaminants and have been reported in wastewater, surface water, groundwater, drinking water, and have also been detected in indoor house dust, and perhaps more important to the current topic, at high concentrations in fracking wastewater. And that's through the work of at least 3 different labs. So we tested a range of various chain length alkylphenol and alcohol polyethoxylates in this other study, published earlier this year in Toxai. And we found that each of these chemicals induced a range of adipogenic activity. So they induced either triglyceride accumulation and or pre-adipocyte proliferation. We then took these chemicals and evaluated them further in a similar way to the previous study that I was just talking about. First, for their ability to activate PPAR gamma, which is this common mechanism often driving fat cell development. So in this case, we found that none of these compounds appear to be active through PPAR gamma, which is similar to the 23 mix of common fracking chemicals and several of the fracking region surface water extracts that also do not. And we confirmed these tests then with a cell-free assay to just be extra certain that we were not activating PPAR gamma. However, several of these chemicals appear to be active through thyroid receptor beta antagonism, another molecular mechanism that can promote fat cell development. This is again in line with some of the results we observed for the 23 mix and other environmental samples that did not activate PPAR gamma, suggesting that this could be a mechanism driving some of the effects on fat cell development observed in this study. And what I should say is that the 23 mix actually also includes both nonylphenol and octylphenol polyethoxylates, which are 2 of these ethoxylated surfactants. So we know that the 23 mix includes these chemicals, so it's kind of a nice tie-in to the previous work. So in summary, we've demonstrated that some chemicals used in or produced by unconventional oil and gas operations can disrupt metabolic health. Notably, this occurred at environmentally relevant concentrations. So even with considerable dilutions of some samples and extracts, we were capable of promoting effects in this cell model. The mechanisms and causative chemicals promoting these effects are an area of further study, and we hope to delineate that further. However, activation of PPAR gamma appeared to be a contributory mechanism for some samples, and thyroid receptor antagonism appeared to be a potential factor for others. The polyethoxylated surfactants detected by several publications in unconventional oil and gas wastewater appear to be active in promoting adipogenesis and also could play a contributory role in some of the effects that we observe here. And lastly, exposure to these chemicals could result in metabolic health effects for humans and animals living in these areas. We demonstrated previously that gestational exposure to the 23 mix induced increased birth weight and body weights in mice. And various epidemiological studies have reported increased or decreased birth weights for children born nearest unconventional oil and gas wells or in areas with the greatest unconventional oil and gas well density. And I'd say that while that may seem a little disparate in terms of having some showing increased birth weight and some decreased birth weight, both of these are associated with increased risks for obesity later in life and bolsters this evidence suggesting potential metabolic health effects of these chemicals. So with that, I'll stop and thank Heather Stapleton and the rest of my lab for the support and encouragement in getting this and several ongoing projects completed. At the University of Missouri, Susan Nagel, my PhD advisor, supported my passion for this topic throughout my doctoral program and encouraged my development of these new research questions. And her lab manager, Jen Cornelis Green, was a great help in much of the sample processing, handling, and coordination. And lastly, this work has really been funded through an R01 and F32 from NIEHS here at Duke. And the samples were collected and produced at the University of Missouri with support from the Passport Foundation, the University of Missouri, and also a crowdfunding campaign on experiment.com. So thanks for listening, and I'll take any questions you may have.
Thank you so much for your presentation. Thanks for, it's kind of a dense topic for someone like me, and you really took time to explain it, and I really thank you for that. And that being said, you did talk a lot about ethoxylated surfactants and how they've been found in multiple studies, and I thought I just wanted to reinforce that they've been found in formations from all across the US and in Canada. And so it's really relevant for anyone living in a place where wastewater, you might be exposed to wastewater. So thanks for that. That being said, also, starting in on the questions, you can go to the question box and follow along, but I was going to read them to you as well. So the first question is, do you have any ideas about other causative chemicals inducing the effects you observed here?
Yeah, that's a great question. And largely, I would say no. So we know the kind of the vast chemical complexity involved in the process. So we know that there are more than 1,000 different chemicals used by industry across the country, between regions, between companies. And I think we've only started to get a handle on what some of the concentrations and toxicity of some of the chemicals might be. So in terms of metabolic disrupting effects, I think that's still pretty much a black box. So I think that to the best of my knowledge, the work that we've done on the polyethoxylated surfactants might be some of the only data that we have in terms of lab studies showing metabolic effects. But I don't think it explains all of it. So I think there's definitely some more out there. It's just going to take some more targeted looking to figure them out.
Okay, thanks. What levels are these effects occurring at relative to the work you've done previously?
Yeah, that's another good question. So most of the work that I did previously at the University of Missouri, so we've concentrated our water extracts to measure endocrine effects. So in most cases, we take a water, we solid phase extract it, so we capture any of the contaminants present on a column, and then elute any of those chemicals out, and we end up with a much more concentrated sample. So anything present in it is concentrated, I think generally about 4,000-fold relative to normal water. So when I was doing most of my PhD work at University of Missouri, we would generally test samples at 40x or 4x water. So any contaminants present in the water were present at concentrations 4 or 40 times more than they would be in pure water. And that's generally where we saw most of our endocrine effects. In this study, most of the effects that we saw were generally actually at levels under that of pure water. So diluting that tenfold further to say 0.4x, so or 0.04x, so up to say a 1 to 25 dilution of what you would have, say, in a stream or in your groundwater, something like that. So at much lower levels in this study than what we have previously published.
So to try to translate that a little bit, you're talking parts per billion, maybe parts per trillion?
Yeah, very likely. I don't know exactly how that would translate, but we're probably at that level, yes.
Okay, thanks. Next question. I don't have a lot of experience with Adipogenesis. Is RXR also involved?
Yeah, absolutely. So RXR is also a kind of crucial player in the process of fat cell development. And in fact, plays a pretty central role. Work that Bruce Blumberg's lab has done in the last year or so has shown that as you transition from the multipotent mesenchymal stem cell into the committed pre-adipocyte or committed lineage of fat cell development, that commitment stage is regulated by RXR. So while it also plays a role in the further development of a fat cell, it also plays a really central role in commitment to that cell lineage. And while we haven't done any testing on it yet, it's definitely a direction that we're interested in going.
Okay, thanks. Let's see, have you ever tested or considered testing drinking water from high production areas?
Yeah, so we have. So we published a paper, I want to say either early this year or late last year, looking at drinking water in Pavilion, Wyoming, and some other areas of Wyoming. So we have tested for endocrine activities in drinking water. However, we've generally stayed away from that for a number of reasons. So a lot of, when you start getting into drinking water, you add a lot of complexity in terms of what are the pipes like inside a person's home, and what is the treatment process like inside a person's home, and what is the drinking water treatment plant process like, and are all those things comparable across your area? In many cases, not. So while it does take a step back from, say, the actual relevance to, like, are people going to be exposed out of the tap to some of these contaminants, it does allow us to make a more broad assessment of a region by not looking at drinking water. But it's definitely something that we're interested in doing, and we've done a little bit of, and I'd like to do more of in the future.
Thanks. Never thought of it like that. So, sorry, next question. Can wildlife also be impacted?
Yeah, so that's a good question, and it's something that I think has really yet to be worked out. So there's definitely a range of studies showing impacts on wildlife. I don't know of any that have looked at metabolic health in particular. And I think that we just need a lot more data in this area. I'd say that we have made some progress in the kind of the lab toxicity assessments through some of the work that we've done and our collaborators have done, but also I'd certainly be amiss if I didn't mention the fantastic work that Greg Goss's lab is doing, showing toxicity and various mechanisms of toxicity for hydraulic fracturing wastewater. And we've also seen a surge in research into the human epi sector. So our health outcomes increased for humans living nearest these drilling regions. But I think where we've seen the smallest amount of work as of yet is the wildlife. So I think that's in many ways, like a lot of this area, we still have a lot of work to do on everything related to unconventional oil and gas. But I think that's probably one of the largest holes, is figuring out, whether wildlife are impacted and to what degree.
Thanks. And we have one final question. It's a good question to wrap everything up. What are some next steps that you have for evaluating this area further?
Yeah, so a few things. So as we alluded to earlier, certainly there are other receptors that play a pretty critical role in this process. And so certainly I think trying to chase down what some of the mechanisms underlying these effects are. So as I showed, we have some suggestion that thyroid receptor antagonism might be driving some of the effects of the polyethoxylate surfactants. But we haven't seen a wealth of evidence in that regard. So there's still a lot of mechanistic chasing to do to figure out even how they're acting, never mind how the environmental samples are acting. And following up on that, just causative chemical identification. So some of the work that we are hoping to do in the next few years is to do some more kind of non-target analysis of wastewater samples, looking for what are these other chemicals out there and what mechanisms, it can isolate specific, chemicals interacting with specific pathways and identify those, trying to work out, what could be not just the chemicals present in the sample, but likely active chemicals present in the sample. And then, like the last thing is further evaluating kind of metabolic health in animals. So as I said, like we showed increased weight gain in the studies that I did when I was at MU. But that's kind of a gross or a crude measure of metabolic dysfunction. And I know that some of the work that Tori Belize and Susan Nagel have continued on is kind of more finely assessing metabolic disruption in the mouse model. So keep an eye out for that work.
Well, thanks. You certainly are going to be busy. I really thank you so much for presenting your research today. And I'd like to thank everyone for listening today. You can learn about future webinars by signing up for our weekly e-blasts on our website at tedx.org, subscribing to our YouTube channel at the Endocrine Disruption Exchange, or following us on Twitter. We have recordings of previous calls and webinars on our website. I'd like to thank you so much again, Dr. Kassotis, for taking your time to present your research, and thank everyone out there for taking time out of your day to watch.
Thanks so much.
Bye.